Okay, good morning, everybody. Let's get started. So I guess last time that we already talked about the glucose metabolism, right? We started carbohydrate metabolism, and we talked about the very important things that we already mentioned, that the blood glucose level, right? Glucose homeostasis, that means that our body need to regulate, need to promote kind of mechanism to regulate blood glucose levels, right, under the very narrow range, under the fasting condition. And we also talked about the how glucose being metabolized in the fat condition in order to maintain the normal levels of blood glucose level after meal, when our blood glucose level increased, and we stimulate two responses, right? The first tier response will be elevated blood glucose level, stimulate insulin secretion from beta cell and pancreas, and then second tier response to elevated blood glucose level after meal is insulin stimulates glucose uptake, right, into the tissues, particularly skeletal muscle and adipose tissue in fat condition, right? When we have a high glucose level and stimulate insulin release, and insulin stimulate glucose uptake into this major tissues. The reason why these two tissues are very important, you guys know which glucose transporter is very important that promote this uptake? No, insulin-dependent glucose transport, right? GLU4. So GLU4 is a major transporter that is response to insulin and promote glucose uptake into two major tissues, skeletal muscle and adipose tissue. So in other words, that in the first tier response, the GLU2 and pancreas are the main, GLU2 is the main transporters, right? And tissue, the beta cell pancreas is the main tissues that involved in the first tier response after meal, right? Does that make sense? And the second tier response, GLU2 is a major transporter, and the skeletal muscle and adipose tissue are the major tissue that involve in the second tier response to the elevated glucose level after meal. So we all talk about this. So and we also mentioned that the insulin release, right? The sequential event of insulin release in response to increased blood glucose level. And so try to remember again, GLU2 is the main transporter that transports glucose from circulation into the beta cell and oxidize through the glycolysis pathway, right, and generate the ADP-ADP ratio. It's increased, that causing the membrane polarization and leads to the increased calcium influx into the beta cell, and eventually you have a high concentration of calcium, which finally triggers insulin secretion. So try to remember this process of insulin secretion response to the high blood glucose level in the beta cell, right? And now we're going to talk a little bit more about glucose transporter into the cells, particularly in the fat condition. Right? We mentioned that insulin is the main hormones that drive the glucose transporter into the tissues. And the important thing is in the both adipose tissue and skeletal muscle, GLUT4 is highly expressed, but not ready, for transport activities. Under the non-stimulated condition or under the low insulin levels, for example, under fasting condition, right? So insulin level will quite low, and majority of the glucose, GLUT4, actually reside inside of cells, not on the cell surface. So it's not ready for transporting glucose yet. Only when our blood insulin levels increase, so insulin, what insulin does is to stimulate GLUT4 translocation from inside of cell to the cell surface. So once you have a lot of GLUT4 on the cell surface in either adipocyte or muscle cells, and then GLUT4 should be ready, right, for transporting glucose into the cells. So again, what insulin does to the glucose transporter is to stimulate GLUT4 translocation from inside of cell to the cell surface, right? So this sort of is the mechanism for how insulin stimulates glucose transporter, by stimulating GLUT4 translocation to the cell surface. And this is under the fat condition, right? When we have a lot of insulin in the circulation, so insulin can stimulate this process. And under the fasting condition, you see the first of all, GLUT4 expression level is much lower compared to the fat condition. And secondly, on the cell surface, GLUT4 is also very few, right? Not many GLUT4 molecules are on the cell surface under fasting condition. So that also makes sense, right? So glucose transport activity is quite low under fasting condition because we already have a lower glucose in the circulation. We don't want to transport, a lot of glucose into the cell. So that will make glucose level even lower, right? So this is something that our body, does not want to do. So under fasting condition, which will make sense, right? You have a lower GLUT4 production and less GLUT4 molecules on the cell surface. And type 2 diabetes patients, the problem is the major problem is insulin resistance. That means resistance means you have a high level of insulin in the circulation, but insulin action is decreased. So insulin cannot efficiently does its function, and in the diabetic type 2 diabetic patients. So insulin resistance again to mean that the cells will produce less GLUT4, and also GLUT4 not able to translocate it to the cell surface, right? So type 2 diabetic patients have problem that they will not have a higher activity of a glucose transporter into the skeletal muscle and adipose tissue, right? So that will leave a lot of glucose in the circulation and causing the high blood glucose level in the type 2 diabetes. So this is the main problem, but also, of course, it's type 2 diabetes is very complex metabolic disease, not just glucose metabolism as a problem, but also lipid metabolism, as a problem as well, because insulin, has a very broad function, right? Not just the regular glucose, but insulin also regulating metabolism. We're going to talk about later. Now here I just wanted to mention the exercise. So in the adipose tissue, we know that insulin is a major regulators that regulate glucose uptake, glucose translocation, right? But in the skeletal muscle, in addition to insulin regulation, the exercise itself, muscle contraction, can also stimulate glucose translocation from inside of the cell to the cell surface. Even you have not very high insulin levels. Right? Remember, I mean, during the exercise, because the muscle utilize a lot of glucose, so actually glucose level are relatively low compared to the resting condition. And also insulin level also relatively low under the exercise. So even under the, I would say, not high levels of insulin, muscle cells will still can uptake a lot of glucose. So because of exercise, the cell can stimulate glucose movement to the cell surface. So the mechanism how exercise stimulate glucose translocation we're going to talk about later, right? Try to remember this is not something related to insulin, but some the very important signaling pathway that involved in regulation of the glucose translocation during the exercise, right? So different mechanism. So muscle has actually two different mechanisms that can stimulate glucose translocation and glucose uptake. One is insulin, right? So during different physiological conditions. Under fat condition, insulin will be the major driver, right, of a glucose translocation, glucose transporter. But during exercise, the non-insulin mechanism is a mechanism, right, to drive glucose transported into the muscle. So this is also tell us why we kind of encourage type 2 diabetic patient to do more exercise, like try to improve glucose conditions, because of exercise, right, can increase muscle glucose uptake. Even you have insulin resistance, even insulin doesn't work very well in the type 2 diabetic patients, but muscle contraction can also stimulate glucose uptake, right? All right. So this I think you guys already done this, right? For informal writing last class. I just wanted to talk about this again. I think that it's very important to help us understand better how our blood glucose levels maintained or regulated. So the different role of GLU2 and GLU4 in glucose homeostasis. Tissue distribution, I would like you guys to tell me, right? So which tissues are the major tissues that express GLU2? Pancreas, liver, right? So liver and pancreas are the two major tissues that express the GLU2. So GLU2 is a major transporters in these two tissues and transport glucose into these tissues. And what about the GLU4? Adipose tissue and skeletal muscle, right? And also cardiac muscle as well. And role in body response to dietary glucose. I mean, for example, under fat condition, when we eat the food, but let's say how GLU2 plays a role in regulate glucose metabolism after meal in the fat condition. So what happened to blood glucose level after meal? Increase, right? And GLU2 we mentioned is what type of glucose transporter in terms of the insulin independent, right? But it's glucose dependent, right? GLU2. So that means when we have a high blood glucose levels, and GLU2 will become active, right, to transport glucose into the two main tissues, liver and beta cell, right, pancreas. So how glucose is metabolized in the liver when glucose is uptake into the liver under fat condition? Remember, we have like a quite a busy diagram, and yes, glycogen synthesis, right? In the fat condition, we wanted to store glycogen, store energy, so we converted glucose to glycogen. So glucose basically is synthesized to glycogen in the liver under fat condition. And what about in the beta cell in the pancreas? When glucose is uptake into the beta cell, what will happen? After meal in the fat condition? Do it again. To stimulate insulin, yes. To stimulate insulin, right? Glucose is uptake into beta cell and metabolized and then to stimulate insulin. Great. So this is what mainly GLU2 does, right? To regulate glucose. So the consequences is that blood glucose level will be reduced, right? So you have a lot of glucose being uptaken into liver and pancreas, and this also we call the response. First or second? First tier response, right? Okay. Now what about GLU4? Under fat condition, GLU4 in the muscle and adipose tissue, right? So under fat condition, when we have insulin levels increase, what would happen to the glucose metabolism in skeletal muscle and adipose tissue in terms of the transport activities? So GLU4, again, in these two tissues, right? Skeletal muscle and adipose tissue. And when insulin levels increase because of GLU2 uptake a lot of glucose into the beta cell and trigger insulin release, so insulin levels increase. And a second tier response will be stimulate GLU4 translocation, right? From inside of the cell to the cell surface and uptake a lot of glucose into the skeletal muscle and adipose tissue. So this is what is the role of GLU4 in the fat condition. And what happened to the glucose when it's taken up into the skeletal muscle in the fat condition? How glucose is metabolized in the skeletal muscle in the fat condition? Fat condition. Anybody? Yes. Well, I mean, in the skeletal muscle. In the liver, you're right. I mean, glucose being converted to glycogen and also to the fat, right? And then the fat can be transported to the adipose tissue and stored in the adipose tissue. In the muscle, similar to liver. Remember, muscle is also the major site for energy storage, right? Glycogen is also stored in the muscle, right? So in the fat condition, glucose uptake into skeletal muscle also converted to the glycogen and stored in the muscle. And adipose tissue basically will have a different role. We're going to talk about it later, right? Also take up glucose, but this glucose mainly metabolized and converted for generation of glycerol 6-phosphate, the triglyceride backbone for triglyceride synthesis in the adipose tissue. Okay, so what happens to glucose homeostasis if GLU2 or GLU4 is defective? What happened to the blood glucose levels if GLU2 is not functioning well? High, right? Yeah. So because what we just mentioned, right? GLU2 transport glucose from circulation into the tissue. If GLU2 doesn't work very well, then you have less glucose being transported into the tissue from the circulation, right? So a lot of glucose remain in the circulation. And also because of insulin secretion in the beta cell also requires glucose uptake, right? So if GLU4 not functioning well, then glucose cannot be uptake into beta cell, cannot be efficiently stimulate insulin. So insulin level will also go low, right? That also will causing the blood glucose level increase because insulin cannot be efficiently to stimulate GLU4 activity, right? So that will also affect GLU4 transport activity in the skeletal muscle and adipose tissue. Does that make sense?
Would
Yeah.
you then say that insulin resistance would be low?
Mean insulin level?
Well, just the sensitivity to insulin.
Oh, insensitivity, right, yes. So insulin resistance, the question was whether GLU4 activity is sensitivity, right? Is lower in the insulin resistant condition, yes. So and GLU4, so similarly, I mean, if we have lower GLU4 activities, then we will also causing the increased blood glucose level, right? And this is mainly, happening in the type 2 diabetes patient. All right, any questions? Yeah.
So is GLU4 dependent on GLU2? So if GLU2 transporters are not working or ineffective, does GLU4 even exist as a secondary response?
Yes. Great question. So remember, GLU4 response depend on insulin, right? So if GLU2 does not function well, cannot stimulate insulin release, right? So insulin action will be decreased. So because again, GLU4 actions depend on insulin, so that will also affect GLU4 activities eventually, right? So GLU4, even you have like you said, have a high levels of GLU4 expression in the cells, muscle, adipose tissue, but the function is low because of lower insulin levels, right? Okay, now let's move on to the glucose metabolism in specific tissues. As I mentioned, tissue different, right? Glucose metabolism, glucose metabolized differently in different tissues. Now let's talk about the liver. Glucose metabolism in the liver. So liver is the most important tissues organ that metabolize not only just glucose but the other macronutrients, lipids and protein as well. So if you see this diagram, it's just it's most comprehensive diagram for glucose metabolism, the liver cell. You will also see the other cells, And so all the 5 different metabolic pathways occur in the liver. So in other words, that glucose can be metabolized through all 5 different pathways in the liver. So for example, liver in the fat condition, glucose being uptake through the GLUT2 into the liver cells and can be converted to glycogen, right? So glycogen synthesis, and of course the most important thing, the first step of the glucose metabolism is the glycolysis pathway, right? So glycolysis and glycogen synthesis pathways, and glucose can also be metabolized through the so-called gluconeogenesis during the fasting conditions, right? So for example, one of the precursors of gluconeogenesis is lactate. So lactate can be uptake into the liver and converted back to pyruvate, and for the synthesis of the glucose, the gluconeogenesis, right? Glycolysis, glycogen synthesis, gluconeogenesis, and also during the fasting, glycogen can be break down and to the glucose and release glucose into the circulation. So the glycogen breakdown pathways. And lastly, the pentose phosphate pathway can also be active in the liver. So again, the liver can do 5 different metabolic pathways, right? Also, again, depend on the physiological conditions, which pathway is more active and which pathways should be, slow down or decrease. And this is the mitochondria. It's a very important organelle, right? That's the oxidative metabolism. So if glucose can be completely oxidized for energy production, then pyruvate, which is a product of glycolysis, right, can enter the mitochondria and convert it to acetyl-CoA and eventually enter the TC cycle for complete oxidation, produce CO2 and water, right? Complete oxidation. So this is the again the glucose diagram showing the glucose be metabolized in the liver. It's most comprehensive, the diagram or the metabolic pathway in the liver, right? So when we learn the metabolism of glucose in a different tissue, and you probably wanted to focusing on first, which metabolic pathway does this tissue do, right? Can this tissue do? And the second thing, you also wanted to try to connect the metabolism with other tissues. So you wanted to try to understand metabolic metabolized flows, whether, for example, under the fat condition, we know glucose actually influx, right, the uptake into the liver cells, and under the fasting conditions, glucose actually released, right, into the circulation. So the direction of the metabolic flow, that will also help you to understand, how liver does the glucose metabolism. And also under fat condition, you see the lactate should be taken into the liver cells for gluconeogenesis, right? Skeletal muscle. So skeletal muscle, if you see, have a little bit less metabolic pathway, right? So that means glucose can be metabolized through some pathway, but not the other pathways. For example, we can see the glucose can be uptaken into the muscle cells in the fat condition, and for the glycogen synthesis, right? So glycogen synthesis activities is high in the skeletal muscle. So muscle cell, in other words, that muscle can also do glycogen synthesis, right, in the fat condition. And under the fasting conditions, the muscle actually release the lactate, so provide lactate, and that will pick up by the liver for gluconeogenesis, right? So it's kind of functioning different, the different tissue cells, and of course the glycolysis. Every type of cells, able to do the glycolysis, right? So glycolysis and glycogen synthesis, the glycogen breakdown pathway is also active in the muscle cells, but you don't see like gluconeogenesis, right? So gluconeogenesis will not happen in skeletal muscle, but the skeletal muscle actually provide lactate for gluconeogenesis that occurs in the other tissues, liver, right? But muscle itself is not the place that, for the gluconeogenesis, right? That makes sense. Any questions? Right? Yeah, of course, TC cycles, complete oxidation can also happen in the muscle cells. Okay, so the question, does skeletal muscle release glucose into circulation? Remember, I think it kind of early, right? We mentioned this earlier. During the fasting condition, liver and kidney are the two tissues that can do gluconeogenesis and release glucose into circulation. And liver is also the tissue that break down glycogen and release glucose into circulation. But we did not put muscle on that slide, remember, right? Why? Why muscle cannot? Muscle has a lot of glycogen, right? But why it's different, from the liver? Why muscle cannot release glucose into circulation? And yes.
Just because it's using the glucose to function or contract, create ATP.
Not quite. Remember, yes.
Does not do blood transporters?
No.
Is one thing?
I think we mentioned the one enzyme, remember? Yes.
It doesn't. Isn't it the muscle doesn't have glucose 6-phosphatase?
Great. Yes. Thank you. Because muscle does not have glucose 6-phosphatase. Glucose 6-phosphatase is the enzyme that converted glucose 6-phosphate to free glucose. Okay? So if muscle does not have this enzyme, then it cannot convert it to glucose 6-phosphate. So glucose 6-phosphate will be further metabolized within the muscle cells, right? Cannot be, converted to glucose. Does that make sense? We're going to learn these enzymes later, right? So this is, again, is an enzyme that converts glucose 6-phosphate to glucose. The muscle lacks this enzyme, so that's the reason why muscle, does not release glucose into the circulation.
What was the name of that transporter?
What's that?
The enzyme, sorry.
Glucose 6-phosphatase.
Okay.
Right? Glucose 6-phosphatase. I think I have. Sorry, no, right here. So, we're going to mention this again right later. So don't worry about this part. But just keep in mind, and muscle lack this enzyme. So although muscle will not be able to directly release glucose into the circulation, but muscle also plays a very important role indirectly, right? For the glucose homeostasis. for example, I already mentioned here, muscle can provide the precursors of gluconeogenesis, for example, lactate, and also alanine, right? Muscle is an important, site for protein metabolism, right? So muscle releases two important precursors of gluconeogenesis, lactate and alanine. So lactate, alanine, that can be picked up by the liver to make glucose in the liver. So this will something like we're going to mention later, called two important cycles, right? Alanine cycles and the Cori cycles. That's how muscle and the liver work together to make glucose. So the muscle basically provides starting molecules, right? The raw materials, and then the liver makes final product, glucose, right? Okay, now brain. So brain, you see the metabolic pathway is very simple. Brain only does glycolysis and complete oxidation of glucose, PC cycles. Right? You don't see any glycogen synthesis pathway or gluconeogenic pathway occur in the brain. Although recently some researchers found brain also does some glycogen synthesis, but compared to liver and muscle, the activity is very low, very small amount, right? But just that the brain mostly metabolize glucose through the glycolysis and the complete oxidation, right, for energy. And also it's very important to know that brain cells heavily rely on glucose for its energy. So glucose is a major energy source for brain cells, right? And again, the glucose usually complete oxidized. Like glycolytic activity is also relatively low in the brain. Once it's converted to pyruvate, and pyruvate will enter again the mitochondria for complete oxidation. Red blood cells is also kind of unique cells and interesting, right? So what's missing in the red blood cells? What organelles are missing? No mitochondria, right? So it's kind of interesting. So the red blood cells, mature red blood cells usually do not have a mitochondria. So that makes the metabolism of glucose in the red blood cell kind of unique, right? So glucose only does glycolysis for ATP production in the red blood cells because no mitochondria, right? You cannot oxidize complete, the product. So the product of glycolysis in the brain, in the red blood cells is lactate, right? Remember so the red blood cells also considered as an anaerobic condition, right? Although you have oxygen, but you don't have a mitochondria. Remember the two important things that are required for aerobic metabolism. One is oxygen, and the other one is the mitochondria, right? So red blood cells metabolize glucose under the anaerobic conditions because of lacking mitochondria, right? Does that make sense? And product of glycolysis is lactate. So lactate produced from red blood cells also is important sources of gluconeogenesis. So if you later we we learned the Cori cycles for gluconeogenesis, the sources of lactate actually from two sources, right? One is skeletal muscle, we just mentioned, and then second one is red blood cells. So it should be very easy, right, to answer this question, why glucose cannot be completely oxidized in red blood cells. Because no mitochondria, right? All right. Okay, so adipose tissue. Adipose tissue, we know the main function of adipose tissue is to store triglyceride, right? And triglyceride usually is formed from three fatty acids molecules and one glycerol backbone, right? And fatty acids, majority of fatty acids is actually from circulation. And initially, either from liver that converted from glucose, remember, under the fat condition, right? And or directly from the small intestine that absorbed it from the diet. So I only put it in the liver, so it should be also from the diet, right? Diet, small intestine. So again, this is happening under the fat condition. So you have a lot of fatty acids being transported from small intestine, initially from diet or from the liver that converted it from glucose, right? And to the adipose tissue. So to provide fatty acids for triglyceride synthesis. And glycerol backbone is normally as a glycerol 3-phosphate is directly, the active molecules that can interact with the 3 fatty acids to form triglyceride. So glycerol backbone or glycerol 3-phosphate basically is from the glucose glycolysis, right? So glucose enter the fat cells is usually metabolized to provide the glycerol backbone for triglyceride synthesis. Most of them, right? Most of the glucose. But also, of course, some of the glucose can be, metabolized for glycolysis and oxidation for energy production. But majority of the glucose enter the fat cells are converted to glycerol backbone, glycerol 3-phosphate for triglyceride synthesis. All right. Any questions? All right. I just try to say this under the fat condition, right? This process, the active. Under the fasting condition, and triglyceride in the adipose tissue is a major storage energy, right, source. So that triglyceride can be break down again through the release 3 fatty acids and glycerol, and these two can be also released into the circulation and picked up by other tissue for energy use. And glycerol is also important precursors of gluconeogenesis, right? So glycerol can be picked up by the liver for making glucose during the fasting condition. So again, multiple tissues that actually work together, right, either during the fat condition or fasting condition to maintain the normal metabolism in our body. And for glucose metabolism, one of the important functions is a purpose is to maintain blood glucose levels, right? Okay, so I think we can stop here and take a 10 minutes break.
Yeah, sorry, yeah.
Like that. That's what I look like.
Get in your backpack and then move that home and you can go to the station at your house. Like oh my god, that's so scary. So this was the ranch she told us that in our living room. And that's a shovel. Like that was this shovel was like this big. It's like it was like this big shovel. Oh my goodness. From another ranch. Look at it. You see like that blood? Oh, you got killed by that. And so they landed. And they put in the house, they put it everywhere. And our shoe, like, so bad. Like, are you bored? And when you and they put up traps, I they never told us this, but they would watch out the traps. And they found another rat on a trap. And they put it. And we're like, what is that? We had a
something that like goes along with grass.
Oh.
I know. Yeah.
I think it's huge. That's what you call a sewer rat. So they came straight into your house.
And then like the next couple days, we see this. Stored in our basement shelf. Look, here's the tortillas. They were all chewed up. And look at all the crumbs on the ground. Literally, that was after we saw the rat. There was another rat. And we're like,
And we have like 30 children, and then they can go back and have 30 more children.
So it was terrible.
So then you got him.
We were like, we refunded.
Yeah.
reflection thing? The what? Did you finish that reflection?
You haven't on Wednesday, you haven't on yesterday.
Your brains don't even matter.
What are you doing? Stop. Ay, ay, ay. Let it rain over you. You will have a conversation.
And then we have flies like so I was at a house and I was working there
Jokes on them. They're gonna find they're gonna find a little rat staring right at them. No, you're actually one down in the basement.
well, we powered so we just and then one day I was at home and it was kind of hot. It was so weird. So I like woke up and there's like 10 flies out of the window and I went to try to find no, it is haunted. It actually is haunted. And I went to and I was assuming
Like, good flies are actual flies.
So I went to the hills and they wouldn't move. They were so dumb and stupid. I could just grab them. They wouldn't fly away. I'm like, this is weird.
Maybe they're babies.
Maybe when they were big. And they were like, and then I went and I was like, yeah. And then I saw it towards like our downstairs, but they were somewhere on those walls, so I like followed them. I was like, this is so weird. I couldn't actually do it. No, it was so annoying. We just came to the house and I was like, and then I was like, because they were so weird. Like, what's happening? I couldn't stop it. And then they were like, and then I just came to the house and pulled up his crib from behind the wall and then like pulled out his fridge and we would have known if there was something. So I went to the basement and there was like 20 on the floor and I was like,
They eat them poison. Maybe they licked it though, and they're all like so
it's a really horrible flies.
yeah, that's so weird.
And so I was like, that's so weird, like, why are there like 30 flies? And then I was like, and then I go back downstairs and I grab my lunch and there's 30 more, like, in the same spot. I'm like, what the hell? I literally, every 3 hours, I was killing 30 rats at a time, or 30 flies at a time. And I was like, this is not I get one.
Why aren't they flying away?
It was so weird. They were, they didn't know if they would like,
It was definitely really plausible.
yeah. And I texted my roommate on it, like, upstairs, they went to the house. I was like, I don't know what's going on. Like, I literally just killed like at least 100 flies in our house. And I don't know where the fuck from. And they were weird and slow and like mentor challenge. And she's like, and I thought that these numbers are garbage, is why like our side door is where our stairs and our kitchen. And I was like, well, maybe it's because we're our garbage. The flies are around the garbage and they're getting into our house. Maybe one fly would get in my open, like not a bunch of them. And then I was figuring in the Anya, the next day, she's like, oh my god, I probably killed 200 flies today. And for mommy, and she's like, my mom and I, like when I got back from work, there was like so many flies, and we were in our basement with shoes, like hitting them in the air, like hitting them on the ground. she's like, I'm going to go eat more ramen. Yeah, and he's like, oh my goodness, they're more and every time me and I would feel confident, I would just speak them up. But after that, she's like, okay, come on, they're not going to feel up because then they'll actually see how their work. And so she's like, come on, like, so then the exterminator they get her laser cam at the beginning. And she's like, yeah, Roman was like, there's only on my course for like 2 days, but it was so weird.
What was the positive?
It was so weird. I was like,
Next grade one though.
they did it last
One time I went in there in the summer too for a bottom of the I would feel like this is weird swimming class. There's no flying away from you.
I would be like,
Cool. Yeah, okay.
Okay. Can you guys hear me? Right. Looks like a battery a little bit low, right? How about the back? You okay? You hear me? Okay. So I think we probably leave about 15 minutes for Dr. Renee Lozen to come to talk about the research paper. So if you guys don't need to take a writing, you can go, right? Okay, looks like you happy. Okay, so now let's start to talk about the specific metabolic pathways of glucose, right? First one is catabolic pathways, for example, glucose glycolysis and glucose complete oxidation, right? So glycolysis and TCA cycles if we wanted to complete oxidize glucose. So let's talk about glycolysis. So glycolysis, as I mentioned, is the first step in the glucose utilization as an energy. And also is the main pathways for metabolism of other sugars like fructose, galactose, and other carbohydrates, right? Not just the glucose. And the enzymatic reaction can take place in almost all type of cells, right? And in terms of intracellular compartment, the glycolysis usually takes place in the cytosol, right, inside of cells, and almost all type of cells in different tissues are able to do glycolysis, right, to oxidize glucose as energy. And some cell types depend only on glycolysis for their energy requirements, for energy for ATP production. Can you tell me one or two cells?
Red blood cell? Or wait.
Red, you said red blood cells, right? Yes.
Okay, great.
Thank you. And of course, I mean, only that means they only utilize glucose as an energy, but not the other nutrients or energy fuels. For example, other energy fuels like fatty acids, amino acids, ketone bodies, right? So the question here is only rely on glucose as energy fuel, cannot utilize the other energy fuels. So red blood cells, right? And brain, they heavily rely on glucose, but brain cells and also utilize the other energy fuels. For example, ketone bodies, we're going to talk about later, right? So red blood cells, it's the type of cells that only rely on glucose for energy. Okay, glycolytic pathways. So this is something I know is not fun, right? Because so many reactions and so complicated. But again, you just try to remember for this class, right? Key reactions and the key enzymes, and how these key enzymes are regulated by hormones, right? Key enzymes or rate-limiting reactions are the same thing, right? Because this rate-limiting reaction, once we activate it or inhibit that, you can slow down or speed up the whole metabolic pathways, right? So why this key reaction is important? Because this key enzyme can control the rate of the whole metabolic pathway, right? So try to remember the key reactions and pathways. So in the glycolytic pathways, we have 3 key reactions. And just for the understanding the purpose, I divided the glycolysis into 2 stages. First stage is called a priming of glucose. So first stage is started from glucose to the glyceraldehyde 3-phosphate, right? Glucose, is a 6 carbon, right? Molecule has 6 carbons, and glyceraldehyde 3-phosphate only has 3 carbons. So in the first stage, glucose is metabolized to generate 2 molecules of glyceraldehyde 3-phosphate, right? The glucose 6 carbon break down into the 2 3-carbon molecules is the first stage. And first stage is the ATP expenditure. ATP consuming stage. That means the first stage, glycolysis does not produce ATP. Instead, they consume ATPs. So 2 steps actually utilize 1 ATP. Right? The first step is, glucose is phosphorylated to glucose 6-phosphate. These reactions utilize 1 ATP. And the second step is the step that converted fructose 6-phosphate to fructose 1,6-bisphosphate, right? And this step also utilizes 1 ATP. So 2 ATP molecules are consumed during the first stage. And in the first stage, there are actually 2 key steps, right? I just put the asterisks here. So first step is the glucose is phosphorylated to glucose 6-phosphate, and the key enzyme is the glucokinase or hexokinase. Why is it 2 enzymes here? Again, depend on the tissues. The glucokinase is more abundant in the liver. So in the liver, the first reaction usually catalyzed by glucokinase, right? And hexokinase is more abundant in the muscle. So in the muscle, glucose is phosphorylated mostly by hexokinase, right? But these 2 enzymes catalyze the same reaction, but just different tissue distribution. So you try to remember this first key reactions: product, substrate, products, and enzymes, right? And the second key reaction is the conversion of glucose 6-phosphate to fructose 1,6-bisphosphate, right? And the key enzyme catalyzes this key reaction is phosphofructose 1 kinase, right? And these 2 key reactions actually consume ATPs. And lastly, generating these 3 carbon molecules, 3 carbon molecules, right? So these 2 molecules can be interconverted, right? So these are all the 2, carbon molecules, but they can interconvert it. And one important thing here I wanted to mention that these molecules, 3 carbon molecules, can be converted to glycerol 3-phosphate, which we mentioned, right, in the last slide. It's glycerol backbones for triglyceride synthesis. So this is sort of the pathways that you can see in the adipocytes, right, from glucose to generate the glycerol 3-phosphate for triglyceride synthesis. Okay. Second stage. So the second stage is started from glycerol dehydrated 3-phosphate, right? This is the last end product in the first stage, remember, right? So again, you're generating 2, right, from 1 glucose. So these molecules enter the second stage. And in second stage, we have 1 key reactions here. It is the reaction that catalyzed by pyruvate kinase that converted the phosphoenolpyruvate to pyruvate. Okay. So the key enzyme is the pyruvate kinase in these reactions. And the second stage is the ATP producing process. That means in the second stage, ATP are produced, right? So you try to get to know where ATP produced, in which reactions, right? So first ATP is generated from these reactions, and this is called substrate levels phosphorylation, right? So this ATP be made by a mechanism called substrate levels phosphorylation. You see the phosphor group, and in these molecules actually contains energy. So breaking down this phosphate and adding to the ATP can produce the ATP, right? Now this is the one ATP molecules that produced from these reactions. And also, the reducing equivalent, reducing equivalent, see here, NADH, right, is made in the first reaction in the second stage. So NADH is a reducing equivalent. Do you guys remember? This is molecules contains energy that can be used for ATP production through the electron transport chain, right? Remember this, right? So, and one molecules of NADH is produced through these reactions. And secondly, the ATP is also can be made in these key reactions, right? So in the second stage, actually 2 ATPs are made, right? From one molecules of glycerol dehyde 3-phosphate, right? That makes sense? In the second stage, 2 ATPs are made from substrate levels phosphorylation, and these are the 2 reactions. And so one molecule of this can make 2 ATPs. And how many molecules actually we generate from the first stage? From one glucose molecules. 2, right? Because these are the 3 carbon molecules. So 2 molecules that produced from first stage enter the second stage, right? So if we thinking about the one molecule glucose, how many ATPs that are made from substrate levels of phosphorylation? 4, yes. 4, right? And that makes sense? Okay. And how many NADH molecules made? 2, right? Okay, sorry. Okay, so if cells or tissues under the aerobic conditions, then pyruvate will eventually enter the TC cycles for complete oxidation, right? But if under the anaerobic condition, for example, in red blood cells, I keep doing this. In red blood cells, because no mitochondria, right? So pyruvate will not be able to further oxidize. So what happened is that the pyruvate will be converted to lactate. And during these reactions, during these reactions, that NADH is required. So NADH will be converted back to NAD, right? Under these reactions. So under the anaerobic condition, NADH will not be made. Does that make sense? Because it made at these first reactions, but utilized in this last reactions, right? So like I go in the circle. Does that make sense? Right? So under anaerobic conditions, the glucose being oxidized to lactate, but only makes 4 ATPs from these two substrate levels phosphorylation, right? You will have no NADH, no NADH molecules are made, right? But under the aerobic conditions, in addition to these 4 ATP molecules, you have additional 2 NADH are made, and then eventually enter the electron transport chain and make more ATPs, right? Do you guys remember how many ATPs can be made from NADH, one molecule NADH? And you guys learn, I guess, biochemistry course probably talk about this, right? How many ATPs? Remember? 2 or 3? 3 or 2, right? Depends. Because this glycolysis occurs in the cytosol, remember? Right? So cytosolic NADH can make either 2 or 3 ATPs, depend on the shuttle system, because NADH cannot be easily transported into the mitochondria for ATP production. So depend on which shuttle that carries NADH into the mitochondria. Right, we're going to talk about later. But other metabolic pathways that in the inside of mitochondria can also produce NADH, and once it's already inside of mitochondria, then NADH will produce 3 ATPs, right? Okay. So something fun, I think. So what happens to glycolysis? Just do some calculations, right? Yeah, I think battery is low. I'm going to change battery. Okay, much better. Right. So now let's do some calculation. First question: Where in the cell does glycolysis happen? Cytosol, right? And how many ATPs can be made from 1 molecule of glucose? So through glycolysis, let's say this is under the aerobic conditions, right? So under the aerobic conditions, the final product of glycolysis is pyruvate, right? And 1 molecules of glucose can be oxidized to, can be metabolized to generate 2 molecules of pyruvate under the aerobic conditions. And how many ATPs are made? Can you guys thinking about it and calculate and tell me? Let's say ATPs under the substrate levels phosphorylation. How many ATPs can directly made? Can be, sorry, 4? Yes. Remember 2 ATP generating reaction, right, in the second stage, and from 1 3 carbon molecules. So from 1 6-carbon glucose should generate 4, right? Remember this. And how many NADH? 2, yes. And to convert eventually ATP, 4 to 6. Remember I just mentioned, right? The NADH made in the cytosol can generate either 2 or 3 ATPs, depend on the shuttle system, how NADH being transported into mitochondria, which shuttle system, right, transported. So eventually it will be 6 to 8 ATP in the under the aerobic condition, right? Okay, so next question. How many ATPs are produced in the aerobic conditions? Anaerobic condition, for example, in the red blood cell. How many ATPs from phosphorylation, substrate levels phosphorylation? Same, right? 4. 2. I'm sorry, I think. Okay. Two ATP. Oh, sorry, I forgot. We forgot about the 2 ATPs are utilized in the first steps, right? So we need to subtract. To take away 2 ATPs that be utilized in the first stage. So that's why net production of ATP is 2, right? Made 4 at second stage and utilize 2 in the first stage, right? So similar for, the last slide, I'm sorry. And under the anaerobic conditions, how many NADH is made? That means 0, right? 0. So, okay. So under the anaerobic conditions, for example, in red blood cells, glucose goes glycolysis to produce lactate, right? Final product is different, lactate, and only make 2 ATP. Okay, so this is something sort of a summary, right? That we just talk about it, The first reactions, first stage, 2 ATPs are consumed, and the second stage, 4 ATPs are made, and net ATP made from substrate level phosphorylation are 2 ATPs, right? Okay. So what happens to pyruvate once it's formed from glycolysis under the aerobic conditions? So pyruvate will eventually enter the TCA cycles, right? TCA cycles for further oxidation. So pyruvate be converted to acetyl-CoA, and acetyl-CoA enter the TCA cycles, right, for complete oxidation to generate CO2, and you have more ATPs or NADH or FADH are produced during the TCA cycles. So, an important molecules I want to mention here is the acetyl-CoA, which is converted from pyruvate, right? So acetyl-CoA is very important molecules that can be used for ketone body synthesis, for fatty acid synthesis, right? We're going to talk about later. And acetyl-CoA also can be produced from lipid, fatty acid oxidation, and amino acids oxidation, right? So it's connect all, the three macronutrient metabolism. Okay. So I'm sorry, I feel kind of not comfortable. Can I sit? I'll try to finish this. So this slide, we're going to talk about the let's see. The other sugars, can also be metabolized through the glycolysis pathway. The fructose, right? So fructose, majority of fructose is metabolized in the liver, right? Because of liver has the very high levels of fructose kinase. And muscle does not have a fructose kinase, but have high levels of hexokinase and aldolase A, right? And liver basically has higher glucose kinase, fructokinase, right, and aldolase B. So as you can see here, the fructose can be directly metabolized to fructose 1-phosphate by fructose kinase, right? And fructose 1 kinase and then be break down into 2-3 carbon molecules by the allose and aldolase B, right? So an important thing is that fructose metabolized bypass these key reactions, right? Remember the two key reactions in here, glucokinase or hexokinase and the fructose 1 kinase. So, and these two key reactions basically is a rate-limiting reaction, right? So if this enzyme's activity is being inhibited, then glucose cannot be, metabolized through the glycolysis pathway. But fructose bypasses these two key enzymes, so will not be controlled by any inhibitors that inhibits glycolysis. Does that make sense? So, and eventually fructose also contribute a lot of glucose, the glyceraldehyde 3-phosphate, and eventually this can be converted, remember, the glycerol 3-phosphate, right? Triglycerol backbone. So when we consume a lot of fructose, a lot of fructose can be directly, metabolized and produce the glycerol 3-phosphate for triglyceride synthesis, and cannot be, regulated by the inhibitor steps. So fructose metabolize generally a lot of glycerol 3-phosphate and for triglyceride synthesis, right? So that is the reason why fructose, high consumption of fructose can produce the, can promote obesity and can promote fatty liver disease, right? Does that make sense? Okay. Okay, this is something I just mentioned, The fructose metabolized bypasses this key reaction, regulated key reactions, and produce a lot of, these molecules and converted to pyruvate and acetyl-CoA, right? And then eventually for fatty acid synthesis. All right. Another thing is about the so-called fructose 1-phosphate. Let's see. Sorry. Oh, yeah. Fructose intolerance. So fructose intolerance is hereditary disease because of the patient's lacks the aldolase, right? And so aldolase, that is very important here, right? Can catalyze the fructose 1-phosphate. So if aldolase B is deficient, then fructose 1-phosphate cannot be break down. So you have fructose 1-phosphate is built up, right? Fructose 1-phosphate is the inhibitors of glycogen breakdown, right? So glycogen breakdown process will be reduced in this patient because of the fructose 1-phosphate is built up. So this patient will have problem that they were not able to maintain blood glucose level during the fasting, right? Does that make sense? Okay. So try to remember, this kind of defects in the fructose metabolism, or why we consume high fructose can lead to obesity, fatty liver disease. Try to understand the mechanism, right? Okay, so I probably just stop here. So if you guys don't have to take writing, and you can go. And if you do, and stay here, and Dr. Rosen will be here soon, right? Yeah.
So for the quiz, we just have up here.
Right. So quiz, just the carbohydrate metabolism, all right? And stop here. Today, everything that we covered in the previous lectures, also including today's.
So mainly the lecture that we want to focus on, what we've lectured through?
Yeah. And also I have a post-study section, study guide, use study guides, right, for your quizzes.
When will they post it?
Moodle?
When?
Oh, already posted. I already posted.
Okay, a question. How do I know if I'm writing intensive or not?
Sorry?
How do I know if I'm writing intensive or not?
Oh, you can ask Dr. Ray.
She's the one that knows if I'm writing intensive or not?
Well, if you have taken other,
Excuse me?
if you have not taken any writing intensive course, then you need write in pencil.
Even if in masters, even if I took them on
Oh, your master maybe you don't need.
if a masters, no?
No, yeah, if you master you probably don't.
Okay.
Yeah, but you can confirm with, yeah.
Okay.
With,
I will ask her. Thank you.
okay, yeah.
Renee, I have a
Yeah.
question.